Hello, everyone. Hope that you are safe these days. I'm glad that you could make it to my talk. In the next couple of minutes, we will talk about an initiative that we started in the beginning of this year when the pandemic started. And I will talk about how we connected the dots and the data silos um, to combat COVID crisis and also combat other diseases. Let me start in March, where there was a challenge published by the White House, and they provided a data set of scientific publications from Bio and medRxiv that were stored together as a zip file. And we thought maybe we could help the pandemic and also help our researchers to get information about COVID-19, to work with the data and structure that. And as I said in the beginning, we just took the zip file, we extracted that, tried to structure the bits and pieces that were titles, abstracts, authors, and so on and so forth. And we wanted to connect the data by the entities of the biomedical field, being genes, proteins, also clinical trials, and so on. And in the end, we wanted to provide a knowledge graph that is publicly available and is free of charge. And within the first week, we could get a team of many different companies and academic research institutions together that all have one thing in common, which is graph technology, and we all work with Neo4j graph technology. And these are the companies here that you see, and also the academic institutions that are working on that project and build it for free, and everything is on a voluntary basis so far. Now, let's get back a little bit to the background, why we are doing that and how we accomplish our goals. When we assume the following scenario, and this is what basically, I guess, everybody of you have, if you're in a conversation with your colleagues or you listen to other colleagues in the conversation, usually they talk about scientific publication and facts that they read in there. For example, here, did you read about this interesting paper about COVID-19 and gene X? And the other researcher says no. And this publication reveals that whatever, XYZ is a marker for diabetes. And the other researchers say, oh, I knew that, yeah. And the other person asked, did you know about protein Y being mentioned to cause lung diseases? And the other researcher said, no, but oh, that's an interesting fact. And you get the point here. In the end, we know a lot of things that we have in our heads, but there are even more things that we don't know and that we cannot connect anymore. And this is because scientific literature is so huge, and we cannot read everything, right? So, for example, in the last 6 months, we had 128,000 preprint publications about COVID-19, which were deposited on medRxiv and bioRxiv. And you would agree that nobody's ever going to read that, but it would be nice to know about the content of them in general. And as I said, we cannot cope with the literature anymore. Problem is that we can also not cope with genes and proteins, with identifiers, with connections between different diseases, different entities, and so on and so forth. So this makes the situation really frustrating, and we thought we could help there by providing technology, providing knowledge, and our knowledge graph with this. Let's get back to our conversation between the two scientists. Usually when they talk and when they would have a white paper, they would draw something like this on the top of this slide here. So gene X is mentioned together with COVID-19. COVID-19 is a keyword in a publication A. COVID-19 is a disease, and also there are maybe clinical studies about that. The other researcher said that protein Y is mentioned together with the lung disease. So this is kind of a rough sketch that we have on the whiteboard. The problem is that sometimes researchers know that gene X encodes protein Y, but usually you don't know that this identifier is that identifier in another database. But what we have are databases, for example, the Ensemble database or NCPI, where we store gene information. And we have another database here, for example, UniProt, where there's protein information. And we have here the pieces of the puzzle, exactly this connection here between the two databases, that a gene encodes a protein. And this is exactly what we do and where we can fill this gap here between the two nodes. And actually, this is true for many, many different genes, for many metabolites, transcripts, proteins, as I said. And we could use that knowledge and bring that together, right? And this is exactly what we try to do. We try to connect the different data silos between genes and proteins, metabolites, pathway databases, and so on. And on top of that, we want to enrich that data and connect it to the literature information that we find in medRxiv, in bioRxiv, in PubMed, and so on. When we think about this core here in our healthcare sector, in the medical field, we have genes, we have proteins, we have single nucleotide polymorphisms, we have transcripts, we have metabolites, right? And usually, as a researcher, you would draw this like this on a whiteboard. And as I said, we extend that by other data sources. We connect everything together and enrich it and annotate these with publications that we get from bioRxiv or medRxiv. In addition to the publications, we also used here in this CovidGraph, we used a subset of SARS viruses patents where we also have text information and annotate also the patents. In the end, this is a pretty busy slide. You don't have to get everything in a second, so I will guide you through that. In the end, we come up with a data model for our researchers that is the same for the researcher and is also the same for our database developer. And this is what makes graph technology really an advantage to other SQL databases. And let me guide you through that again. So the yellow part here was 128,000 publications coming from bioRxiv and medRxiv, dealing with only COVID-19. We are now at a stage where we integrate all the 30 million PubMed articles. We structure the data sets so that you can distinguish between citations, abstracts, authors, their affiliations, and so on and so forth. We connect this public text information to the 30,000 patents coming from the SARS and MERS coronaviruses. And as I said, we are connecting these to the biological entities that we have in the biomedical field, being genes, transcripts, so RNAs, proteins. We connect the proteins to their gene ontology terms. The genes, for example, are also members of metabolic pathways. We have information from 55 human tissues coming from the Broad Institute, where we know that a specific gene is expressed in a specific human tissue. When we go to the lower part of the slide, we have nearly 2,000 clinical trials dealing with COVID-19, and we have an API that pulls more data into our CovidGraph from clinicaltrials.government. And in the end, we have the case numbers here from Johns Hopkins University and the United Nations. So what are we going to do with the text data? As I said, nobody's ever going to read that stuff. So we try to help our researchers by named entity recognition, where we can classify that specific words are recognized and classified as, for example, a gene, or this is a eukaryote, or this is a physical science term here. So we analyze the text automatically by using natural language processing, named entity recognition, and we convert that directly into a graph. And since many of the keywords are mentioned all over again, we can connect these different entities. In the end, our long-term goal is not only to provide a data source and an information source for COVID-19, but also for other diseases. And this is because many different diseases are connected to COVID-19. For example, diabetes. We know that several people have an increased risk of getting infected, and these are diabetic patients, and they also have an increased risk to die. So it would be nice to know more about this hidden connection between COVID-19 and diabetes. Now, this was the introduction to the database itself. As I said, everything is publicly available via CovidGraph.org. Now, the question to you as user is, how can you connect to the database? How can you work with the data? And we have several different applications from our partners that collaborate with us, and we provide that to you, and you can choose between different applications here. And just to mention a few of them, we have a Neo4j Bloom instance here. We use Linkurious for several queries. If you are a database developer, you would like to use Cypher as a query language directly to Neo4j. And on the lower left, we have our Visual Graph Explorer where you can interactively explore the data without knowing anything about computers and Cypher queries. So I would use the rest of the time to show you 4 different use cases, and I will show you only screenshots because, as you might know, that live demonstrations never work if you want them. So we stick to the screenshots, and believe me, it works in the end. You can try it out yourself. Let me start with the first use case, a very simple one. What is the most relevant gene in COVID-19? Pretty simple question. It's not that easy to ask. But what we have is scientific publications and the genes that are mentioned in these publications. And we run the Graph Data Science library from Neo4j and ask the question, what gene is mentioned in all of these publications? And the result is we find ACE2 mentioned most in most of the publications, which makes totally sense because this is the receptor the coronavirus uses to enter the cell. Interesting point here is that the end of your query that you do on the database is just the beginning of your interactive data analysis. So you can drag around here these nodes and these edges. You can hide them. You can click on the publications here in blue and interactively explore that. And as I said, this is especially helpful for people being medical doctors or researchers that are non-computer scientists that cannot deal with Cypher queries and Python scripts and so on. You can do similar things in our Visual Graph Explorer, as I said. You can type a gene on the right side here. You can type in a gene or a paper or an author here. You can search for that. You can search for patents. And once you click on one of the nodes here, you will find the information, for example, here for a publication, an abstract, the reference papers, the authors, the mentioned genes, and so on and so forth. Second use case, find a tissue-specific gene in publications. As I said, we integrated human tissue information where we have the expression data for each and every tissue. And here you see an example why Neo4j and also the Cypher query language is so easy to understand if you are able to read that. We have here the data model where we start from a human tissue where a gene is expressed. This gene is mentioned in an abstract which belongs to a paper. And as I said, we want to filter that to a lung-specific gene. So what we do here is we match a tissue, we specifically filter here for lung, and then we type in here expressed in a gene, and the gene should be mentioned in a paper in yellow. And so Cypher as a query language is pretty easy to understand. Everything what's in parentheses is a node. Everything what's in brackets is an edge. And this is how you build your query. Next use case. We have several patent lawyers that are researching patents and scientific publications for the one or the other company. And one of the questions here in our test case was, find me the molecular pathway that Novartis is working on. I show you here a new application that we have from the Derivo company from Germany. So we are on the same database. We query here our entities being the companies where we filter here for Novartis, and you see for the COVID-19 or COVID in general, COVID patents, Novartis has 40 patents so far. And I did the query here through the whole graph, and this is what I've done in 30 seconds manually. If you type in a query, it takes you 0.2 seconds. And we query from the patent owner via the patents, via the genes that are mentioned in these patents. The genes are connected to their transcripts. The transcripts are coding for a protein, and the protein is connected to the function in gene ontology. And this is what we see here on the lower part. You see this little node here in the end, and we see that regulation of heart rate and cardiac muscle, cell cardiac muscle regulation is a molecular term in a pathway where Novartis works on. So pretty interesting, isn't it? The fourth use case was done together with a company called Meltwater. And they have an interesting data set because they are scanning press releases in all different media. And I was interested in press releases about clinical trials about COVID-19. And once I saw the database and once I saw the data, I also saw a property that they calculate, which is a sentiment of a text. So is it a more positive, a neutral, or a negative text talking about a specific topic? And I was interested in the clinical trials, as I said, if some of them are have keywords that are more positive or more negative. And what I saw is here that also we have a named entity recognition on these texts where we automatically recognize drugs as hydroxychloroquine, remdesivir, and so on and so forth. And I did a clustering analysis and clustered all the clinical trial texts here in these yellow, red, and green nodes. And the light blue, so the cyan nodes here are hydroxychloroquine, and the deep blue nodes are remdesivir. And you can clearly see the sentiment of hydroxychloroquine together with COVID-19 seems to be more neutral, or rather a little bit negative. Where on the other hand, we have texts talking about remdesivir in clinical trials that are rather positive. So this was quite an interesting use case, and I leave it as it is, just showing you the power that we have and that you can query in the graph. I hope that I could show you that CovidGraph is a nice repository where we structure, analyze, integrate, and connect data from different data silos that are generally highly connected, but as I said, they are data silos. And I hope I could show you that we connect everything together in one database so that the researchers do not have to swap between databases. I showed you an example how we are annotating and enriching the text data by using natural language processing and other machine learning methods. Um, I showed you that Neo4j as a graph database is an amazing tool for storing this data, especially if it is highly connected and very heterogeneous. As I said, the database is one thing. The different thing is on top of the Neo4j database, we have several different user interfaces depending on what preference you have and what user you are. And I showed you for use cases just some examples from our daily work here that could be valid in your medical research in your company or in academic institutions, but also in intellectual property. Everything what I showed you so far is open source. It's publicly available. You can also download it on CovidGraph.org. And in the end, I just wanted to mention it again. We are at a stage where we integrate more and more data, which goes beyond COVID-19. So we try to help researchers in order to understand connections between different diseases, which can be COVID-19, which can be cancers, Alzheimer's, and so on. And with this, I'm finished with my talk, and just wanted to show you the last slide with the team here. So we are a team between 15 and 20 people that are all working on a voluntary basis, and we have a lot of fun. We learned so much and get a really, really nice attention during the last month. And with this, I'm at the end of my talk, and I thank you for your attention, and hope to see you soon live in the next conferences. Bye-bye.